He didn't want to unwind. What am I doing for Halloween? Y'all really want to know? I am dressing up. I'm actually dressing up. So y'all know I got a puppy, right? A puppy. I have an 80-pound, 6-month-old Great Dane. She's legitimately 80 pounds. So I got a Velma. I got stuff to be Velma. And she's going to be Scooby-Doo. And then my son is going to be, it's from How to Train Your Dragon. Hiccup Horrendous Haddock the Third, the dragon rider. And he's got a little stuffed animal of the dragon. It's cute. Okay. So it's going to be fun. But why they couldn't put it on Saturday rather than hype up the kids on sugar on Sunday night and let's send them to school on Monday? That just seems like a bad decision. We should change it. Like Thanksgiving is the last Thursday in November. Halloween should be like the last Saturday in October. Can we vote on that? Okay, so make sure you do your seating chart. Make sure you do your seating chart. And then we were on, I think this. Yeah, we were talking about functional groups, right? Yes? No, maybe so. Yeah. Okay, so we said there were 3 different common mechanisms that enzymes use. What were the 3? Acid base, covalent, and metal ion. Okay, let me put my chat. I forgot about my chat. Okay, and then there are 2 different kinds. We're going to first talk about acid base. There are 2 different kinds of acid base catalysis. One is general, which refers to what? Any amino acid side chain acting as an acid or a base, right? And then what was the specific? It's water. Water. Okay. So we said that these are different amino acids that can have both an acid form and a base form. And so what we're going to see is in the mechanism that they can behave as either. Okay. And I really wanted to do this on my iPad, but I couldn't get it to connect. I don't know why, but I really want to be able to zoom in because it's pretty small. So I'm hoping that y'all have this either printed out or can pull it up on your computers or something. But okay, so acid-base catalysis. We're gonna talk about this is a general acid-base catalysis mechanism, and this happens in a ribonuclease enzyme. So if it's a ribonuclease, what does it do? Ribo for RNA or DNA? RNA. Okay, so we're going to do something to RNA. It's an enzyme that's going to do something to RNA, and it's going to degrade RNA. Okay. So we have a nucleic acid, and we're going to try and break off individual residues from that nucleic acid, right? So this is our example of using acid base. So here's our RNA molecule. RNA molecule is a phosphate, a ribose sugar, and a base, right? That ribose sugar is going to be connected to another phosphate, which is connected to another sugar, which is connected to a base. So do you see the backbone of the RNA? Yes, right? Okay, now, then you also need to look at the residues that are inside. This is the enzymatic pocket of the enzyme. So this is the ribonuclease enzymatic pocket. And we're going to look at His 119, and we're going to look at His 12, right? So remember, His can act as either an acid or a base depending on the pH, right? So if we look, let's look first at His 12. His 12 is going to act as a general base. If something is a base, what is it going to do? Is it going to give up a proton or is it going to take a proton? It's going to take a proton. Okay, so do you see on this carbon right here, we have a hydroxyl group. So His 12 is going to steal this proton from the RNA molecule. Once that proton is stolen, this oxygen is now going to go in and attack the phosphate, okay? When that happens, His 119 is going to act as a general acid, and it's going to actually donate its proton. So when we look at what's the result of that, what happens? You're going to lose this bond, right? You're also going to lose this bond right here because the phosphate is now going to make a new bond with this oxygen. This is really hard to see. Can y'all see that? So this phosphate is going to make a new bond with that oxygen, right? And that's what we see here. This phosphate has made the new bond with the oxygen. So if it does that, it can only have so many bonds, right? If we take one away, if we add one, we have to take one away, right? And so what we're actually going to end up doing when we do this is we're going to break that bond right there. So what's going to happen is we're going to release one base from the RNA, right? So we're going to chop it up one residue at a time. So now His 12 is going to act as a general acid. So here's His 12. This is the same residue. It's going to act as an acid. It's going to donate this proton back over here to the RNA molecule that's still in the enzymatic pocket. We got rid of one. So this is a 1 by 1. We're going to break down the RNA molecule. So we're going to give that hydrogen back, right? And then His 119 is going to act as a general base. It's going to actually accept hydrogen from water, because remember, this is an enzyme. So whatever it does at the end of this whole pathway, we have to be back to our original state, right? So if you start giving up protons and taking protons, if you stay in that state, you're not regenerated. So you have to remember that in an enzymatic pathway, that's what this is. You're going to have some things that are facilitating the catalytic mechanism. And then you're going to have some parts of the process that are there to regenerate the enzyme, because we want to get back to our original state. So our His 119 is going to steal this proton, right? Because what did it start out with? Did it start out protonated or deprotonated? Started out protonated. So we need to get it back to the protonated state. So it's going to take a hydrogen away from water. It's going to reform that hydrogen attached to the nitrogen, and then we're going to have a hydroxyl group, right? We'll have left an OH. That hydroxyl group is actually going to attack the phosphoryl group that's on the rest of the RNA that's still in the enzymatic pocket. And so when it does that, now we end up with a phosphate group that has a hydroxyl. We have our His 119 that is protonated, and we're back to our His 112 being deprotonated. So we're back to our original. So then what's going to happen is this RNA molecule is going to just shift down inside the enzymatic pocket, and we're going to start all over again, right? So what kind of catalysis do we see in this process? Remember our three that we just listed at the very beginning of class? We have general acid base, right? So the general acid base, who's acting as a general acid and base in this scheme? His 119, sure. So here it's acting as a general acid, right? What about in that first step? What is His 12 doing? Acting as a general base, as I like try to underline it and cross it out completely, as a general base because the thickness is so dark, right? Now, His 112 starts out as a base, but then what does it do in the very next step? It acts as an acid. And that makes sense, right? As soon as you can pull a hydrogen off and attach it to His, now that His can act as a general acid, and it can give up that hydrogen. So a lot of times what you're going to see is whatever residue is participating in that catalytic activity, it goes back and forth, and it can act as either. Okay. And then the same thing in His 119, it acts as a general acid, and then over here now it's acting as a general base. And you need those opposite things happening in order to regenerate all of your active site inside of your enzyme. So now we're back to our original conformations. Everybody who had a proton has a proton. Everybody who was deprotonated is deprotonated.
Oh,
Sure.
oh, so what's the purpose of this?
So the whole purpose of this is to break down RNA, right? That was the ribonuclease. We want to degrade RNA. So we want to pull off individual monomers from our RNA one at a time, right? Because is RNA going to stick around in the cell for very long? Do you want it to stick around? No, it's not like DNA. DNA, once we make it, we need to keep it. RNA is unstable in and of itself, but you also want to have processes there to degrade it so that if you want to upregulate expression of whatever gene and the environment changes, you need to shut that down as quickly as possible so you degrade all the mRNA that's sitting around in the cell. Okay, yeah.
Since we use water as a way to break it down, would you call this hydrolysis, or could you at least compare it to hydrolysis?
Well, so it's not hydrolysis. It's not hydrolysis because you're not using the water to break the compound up itself. You're using it just to reprotonate the side chains that are active in the catalytic mechanism.
I just saw it.
But yeah, no. And water can do a lot of different things, and it's very important for a lot of different mechanisms within the cell. Yeah. So let's look at a few more. And I wanted to say, all these mechanisms that we're going to cover in class, what I did for y'all is I also posted them on Moodle without the descriptions. They have the descriptions in your book, right? You can always go and look at the descriptions. But like if you want to be able to look at it and go, which residue is acting as a general base, or where is this one acting as a general acid? You know, you can have the blank one there and try to identify those characteristics on your own and then go and check it in the book, right? So you have all of these mechanisms blank. It's like under all the animations and stuff, okay? So that you can go in and you can look at them specifically. All right. Another type of mechanism is covalent catalysis, right? So when we talk about covalent catalysis, we're always talking about things that are nucleophiles and electrophiles. So a nucleophile is an example of a molecule that is electron rich, right? It has lots of electrons. Something that is an electrophile is electron deficient. D-E-F-E-C-I-E-N-T, deficient, I think, right? So in this case, what we're looking at in this mechanism is we have, what do we call NAD, and NAD plus and NADH? What do we call that? A did I hear it? Coenzyme, not a cofactor, right? Because a cofactor is like a metal ion or something like that. It's something inorganic. But NAD plus NADH, that is something organic. So that is going to be a coenzyme. Okay. So in this case, right, what we're doing here is we're looking at not acid and base, right? We're not going to be talking about transferring hydrogen specifically. So when we see our substrate, this is, for example, our substrate, we have a carbonyl carbon, right? And a hydrogen attached to that carbonyl carbon. So when our substrate comes in, we're going to initiate formation of an intermediate. So the intermediate, when it comes in, let me start over. The substrate, when it comes in, is going to interact with the active site. But when do we have the greatest number of contacts between the enzyme and the substrate? When it's in the substrate form or when it's in the intermediate form? Intermediate form, right? That's when we say that affinity is the highest. That's why those intermediate analogs have had such high affinity. So if you kind of remember that, you're gonna realize that. So when we have our substrate come in, what's actually going to end up happening here is that these electrons here on this sulfur, right, are going to attack the carbon. When that happens, Now this oxygen is going to be very negative, is going to go in and steal this hydrogen. So let's see if I can move this. Say it again.
We have to look attack on the carbon and then
Mhm.
electrophilic attack on the hydrogen.
On the hydrogen, exactly.
Or not.
Yes. And those are covalent interactions, right? So now what do we have? Now we have the carbon. This was our initial substrate that's actually bound to the sulfur of cysteine, right? So now we have this unstable intermediate, right? We're going to actually make a couple of intermediates. Do you remember those little energy diagrams when we saw those multiple humps? Each hump corresponds to a different intermediate. And that's what we see in these pathways, that you're not just going to make one intermediate, you make a couple of different intermediates, right? So then in this case, we need our coenzyme. Our coenzyme of NAD+ is actually going to oxidize our intermediate. And so what we see, what happens in an oxidation reaction? Remind me. Oxidation is gain of oxygen or loss of electrons, loss of hydrogen, right? So what happens here is that this hydrogen that's attached to my carbon is going to get transferred to the NAD+. We're going to form NADH, right? Now, at the same time, when that carbon loses its hydrogen, it's no longer going to have 4 bonds to it. So what it's going to do is it's going to convert that oxygen single bond to a carbon oxygen double bond. So we have another intermediate, right? Now, we have to, a lot of these cofactors have to be regenerated. So right now we're sitting and we have NADH. NADH cannot participate in the rest of the reaction. So the NADH is actually going to get swapped out. NADH gets kicked out, and we pick up a new NAD+ molecule, right? So this positive charge on this new NAD+ is going to stabilize our negative charge on the carbonyl oxygen, right? Because once you have that double bond, you're going to have a partial negative charge on the oxygen, partial positive on the carbon. Now that you have a partial positive on the carbon, you're actually going to interact with a negative charge on a phosphate group. So what are we doing in this process? What happened? If I'm taking a phosphate group and I'm putting it onto a carbon group, what am I doing? What's that process? Phosphorylation. This is a general phosphorylation reaction. And we think of it as you just add a phosphate group, but it's a stepwise process. It doesn't happen all in one step. So our carbon's partially positive, going to interact with partially negative on the phosphate group. And then we still have, so you would actually associate your carbon and your oxygen at this point to phosphorylate it. But we want to disconnect our product from our enzyme. So that's the very last step, where the sulfur is actually going to interact with the hydrogen on a nearby histidine residue. And it's going to take that hydrogen so that we can now have the sulfur bound to a hydrogen, and we can release our phosphorylated product. Yes?
So in those last 2 steps, the positive NADH+ just kind of stabilizes the negative oxygen, so that way the rest can happen without the oxygen interference.
Correct.
Like ionic or polar stabilization.
Correct. And because you've now replaced the NADH with the NAD+, now you have the correct starting material again as well. So it's kind of a twofold thing. You're going to have to swap it out in order to get back to normal, but you also need it there to stabilize the negative charge on that oxygen. Yeah. Doing okay? I know it's a Friday. This is hard on a Friday. But this is all really based on organic chemistry, right? So the things that you've learned in organic, hey, you get to apply it. Isn't that awesome?
The proton transfer.
Yes, yes, yes. It's so awesome. All right, metal ion catalysis. Y'all are like, no, this is not that exciting, Dr. B. So when we talk about metal ions, what's going to be the charge on metal ions in general? We're going to have a positive charge. Okay, so we're going to use an example. If we have a metal ion, is that going to be a coenzyme or a cofactor? This is going to be a cofactor. Okay. So you can have one of two things happening with metal ion catalysis. Sometimes the metal ion is basically stuck, covalently attached to our enzyme, and sometimes it's just associated with it, and it can kind of come and go, right? So you actually have two different kinds. So if you have it covalently bound metal ion, so this is going to be a metal activated. Oh, no, I'm wrong. If it's tightly bound, it's a metalloenzyme. And then if it's just loosely associated, this one is going to be your metal activated. Each time, it doesn't matter which one you are. If you don't have the metal ion, your enzymatic process is not going to happen. Enzyme. But sometimes it's covalently bound and it's stuck, and sometimes it's just loosely associated. But either way, you need it in order to work, right? Okay, so in this case, right, we have zinc here, and it's associated with 3 hisses. So in this case, if this is a zinc 2+, what do you think the charge is on these histidines? They're going to be negative, right? All right, so what we're going to have here is we're going to have a water molecule that's going to come in, right? And so the water molecule is going to associate with our zinc because the oxygen is carrying a partial. Does that show a positive?
It does show a positive.
That is a partial negative. It's a partial negative. So we're associating our positively charged ion with our negatively charged oxygen, right? So what's going to actually end up happening is we're going to lose one of those hydrogens off of the water. So this oxygen now has two electrons, a lone pair of electrons, and it's carrying a negative charge. And so here we go. Now we can take carbon dioxide. What do we produce as we undergo cellular respiration? Carbon dioxide. So this is something that's going to happen a lot in our blood.
Maybe it's a positive because the oxygen is bound to the same instead of having to put it in a lone pair.
Maybe it should. Well, I guess they could say it that way. But it doesn't. It should. But I don't like the way they do that. I don't like the way they do that. If that's what it is, okay. But I don't like the way they do that.
Show the partial negative before.
Before, yeah. Otherwise, it wouldn't associate and you wouldn't have it in the first place. All right, so we've made carbon dioxide, right? And we have this oxygen over here that's very negative. Why is this oxygen able to hang out in this state? Because it's associated with a positively charged zinc ion here, right? So the whole point of having positively charged ions inside of our enzymatic mechanism is that we want to stabilize our intermediates. We want to promote the proper orientation. We want to stabilize anything that's really highly charged. A lot of times that's what you find in these reaction mechanisms, is that the intermediates have large negative charges and therefore they need some kind of positive charge to balance it out. All right, so now our negatively charged oxygen is going to attack our carbon. We're going to create a bond between the oxygen and the carbon. That carbon cannot maintain 5 bonds. So what's going to happen, we're going to lose one of these double bonds and our oxygen becomes negative, right? Now what happens if we bring in a new water molecule, what we're going to do is we're going to kick out this new substrate, this new product, I guess, to say it better that way, this new product, and that new product is bicarbonate. So this happens in our bloodstream all the time, right? All the time. But you have to think about the main reasons why you're going to have a metal ion there. You want things that come in negatively charged to be oriented in the proper way so that catalysis will begin. Then once catalysis begins, you're going to make these unstable intermediates and the positive charge will stabilize those intermediates. So it's kind of like a twofold a twofold process that's happening. So a lot of times when we have coenzyme dependent reactions, like what we talked about with NAD plus and NADH, right? A lot of times you find those in dehydrogenase reactions. And when we're talking in the body, we do have NAD plus and NADH, but those aren't the only ones. You also have NADP plus and NADPH. Then you have FAD, FADH2, and FMN and FMNH2. You do not have to memorize the structure of all of them. They're large structures. So usually what we do is we look at just the part of the structure that's really important to us. And then what do we do with the rest of the molecule? We call it R, right? So that we're only looking at the part of the molecule that's actually going to participate in the enzymatic reaction, right? So if we look at NADH, if we convert it from the reduced form to the oxidized form, our reduced form has an additional hydrogen, right? So we have, when we convert, we lose that hydrogen right here, and we turn that ring into a ring with 3 double bonds. And what happens to our nitrogen? Our nitrogen becomes partially positive. So a lot of times you're going to use this inside of the body to undergo dehydrogenase reactions, right? So here's our example. You're actually going to do this in lab. Lactate dehydrogenase, LDH, is an enzyme you are actually going to purify in lab. And you're going to look at the activity of lactate dehydrogenase. And so you're going to actually monitor this exact reaction happening. So lactate dehydrogenase will convert pyruvate into lactate. So it will actually donate its hydrogens to the pyruvate in order to create lactate. And we'll talk about in later things why that's really, really important and what role that plays. But it's not the only time that we use these coenzymes in dehydrogenase reactions. We have lots of other dehydrogenase reactions, like succinate dehydrogenase. That is an enzyme that uses FAD, and the FAD will actually pick up 2 hydrogens from succinate, and it's going to make fumarate. And these are substrates that you probably are not familiar with yet. But when we get to glycolysis and we get to the citrate cycle and the electron transport chain, you are going to get super familiar with them. So don't be afraid that you're like, oh my gosh, I don't know. You don't have to memorize the reactions. That's what I'm telling you. Yet. You don't have to memorize the reactions yet. All right. You can also have transformation reactions, right? So a lot of times, you're going to see some isomerization reactions. Remember we talked about isomerization before? How do you know something's an isomerization reaction? You end up with the same what? Molecular formula. So look at dihydroxyacetone phosphate. If we write the molecular formula for it, after it undergoes a conversion with triose phosphate isomerase, we have the exact same molecular formula, but now we have glyceraldehyde 3-phosphate. So basically, we've just rearranged the atoms inside of the molecule. You're also going to see condensation and dehydration reactions. So here in this case, right, we have something that has 2 phosphoryl groups attached to it. And we have another molecule that has 2 phosphoryl groups attached to it. So what I want to do is I want to take these hydrocarbon ends and I want to link them together. So basically what I'm going to do is I'm going to take the 2 phosphate groups off of one of the molecules and then replace it with the hydrocarbon of the other. So what's going to happen in our transferase reaction? What are we doing? You're just transferring. That's why they call it a transferase. You're usually combining molecules. You're usually sometimes adding a phosphate group. This is removing 2 phosphate groups, but sometimes it's adding a phosphate group. But it's always when you want to usually build a molecule, you want to add things to it, right? Okay. You can also have reversible covalent modifications. Now, reversible covalent modifications, this is our molecular switches. These are things that turn enzymes or turn, usually it's enzymes, but turn things off and on, right? You want something that is reversible because you want it to be like a light switch. You don't want it to be a permanent change and then you're done, right? You want to be able to still respond to the environment and respond to cues from that environment in order to, let's say, produce insulin, right? If I've just eaten, I want to make insulin. But as soon as my blood sugar drops below a certain point, do I want to still make insulin? No, because my blood sugar is getting too low. And your brain uses glucose as a primary energy source. So if you run out of glucose, your brain is not going to function anymore. And so you can't get below a certain glucose level inside the bloodstream. So you need to be able to turn that insulin expression off, right? So this is an example of this. This is how we turn insulin signaling off and on, depending on whether or not this oxygen right here is phosphorylated or not. If it's phosphorylated, then insulin signaling is on. If we go through in this direction, a phosphatase, we're going to remove a phosphate group, right? So in one direction, we have a phosphatase. In the other direction, we have a kinase. A kinase is going to add the phosphate group, right? So kinase, you can say plus phosphate, phosphatase is minus phosphate. And where do you get the phosphate from? Typically, ATP, typically from ATP. It's not the only source, but it's a common source. Okay. All right. So now we get to talk about reaction mechanisms. So we're going to use some example enzymes and look at their reaction mechanisms, and we're going to see lots and lots of weak non-covalent interactions. And it'll make sense when you draw on all of that organic chemistry knowledge that you maintain, that your professors are going to be so happy that you remember. So when we talk about all these reaction mechanisms, right, we want to be careful that the substrates that are coming in are going to be oriented in the correct way to interact with our amino acid functional groups, right? So we'll have part of the molecules that'll help with that orientation, and then we need to put them in proximity to the reactive centers, right? So we're going to really focus on those reactive centers today. So here's another example of an enzyme that will break things down. It's called chemotripsin. Chemotripsin is a serine protease. What does that mean? It's a serine protease. It means it won't ubiquitously cut up proteins. It's going to be a little bit specific. It's going to cut up proteins that have what? A serine in them, right? So you'll see lots of different proteases, and each of the proteases, I'm going to skip ahead real quick and I'm going to show you something. Each of the different proteases, here's chemotripsin, here's trypsin, here's elastin, have unique enzymatic pockets that facilitate cleavage of particular kinds of molecules, right? So here we have chemotripsin. Chemotripsin is going to use a serine in order to break up certain residues within our protein. So what do you see here? An aromatic ring. So serine proteases cut at aromatic rings. So trypsin, on the other hand, has, what is ASP? What is ASP? Aspartic acid. Aspartic acid is what? Negative. So it's going to want to interact with positive. So it's going to cleave proteins that have positive residues, like, for example, arginine, right? Elastase is one that is actually going to cleave elastin. And elastin is very, very flexible molecule. That's kind of why they call it that way. And think about proteins that have like big side chains, right? They're kind of restricted in how they can orient in 3 dimensions. So if you had a protein that had just little bitty residues like alanine, right? It'd be very flexible. So the enzymatic pocket in elastase is going to accommodate only really, really small residues. so that each of the individual enzymes are specific for their targets. Yeah? Okay, so let's back up. All right. So chemotrips, and we call it a serine protease because serine is the catalytic amino acid that facilitates the cleavage. So you actually have 3 residues within a serine protease that are important. A histidine, what is ASP again? Aspartic acid, and we have serine. And these are going to undergo hydrogen bond formations in order to break down the protein. So in the very beginning, you have these amino acids interacting with one another before your substrate even comes in. So we have ASP102. So we have a negatively charged oxygen, right? We have His57. His has a hydrogen, partially positive hydrogen that can interact with that oxygen, right? That's a hydrogen bond. Then we have on that same histidine a nitrogen. This nitrogen is partially negative, so it can interact with the hydrogen from serine, right? Because that hydrogen is partially positive. So what actually ends up happening is because we have aspartic acid that's interacting with this hydrogen, this nitrogen is going to come in and steal a hydrogen from serine. So now histidine is protonated, right? And it's still interacting with aspartic acid. So we're like priming our enzyme. Now this I want to zoom in on, and I don't know if it's going to let me do that. Zoom. Let's go as big as we can. Oh, I'm going to move that here. Okay. Well, that's better. Okay. So if we look at our catalytic triad, we're looking at 3 residues, right? ASP102, His57, and serine 195. These are the catalytic enzymes. Now, catalytic amino acids. Now, there are other residues that are important in stabilizing our intermediates and things like that. And one of those is glycine 193. So we have a polypeptide that comes in. Remember I said chemotrypsin likes to cut amino acids that have a what? That have a ring structure. So that ring structure is actually going to fit perfectly into a pocket. But this is not a catalytic part of the enzyme. This is just orienting your substrate in the right way. So our substrate is going to come in and it's going to bind inside of that pocket. So when it binds inside of the pocket, the first thing that's going to happen is this is, if you go in, if you've got this on your notes, go in and name this. This is our His57, right? So His57 is going to remove a proton from our serine 195. This is serine 195, just so that you can keep track of the residues, right? So our His is going to steal the hydrogen from the serine. That's what we saw in the last slide too, right? So now we're going to be able to have nucleophilic attack from this oxygen on our carbon in the backbone of our amino acids, right? So it's going to actually attack the carbon that's involved in the carbonyl bond. So now that oxygen fits perfectly in the oxyanion hole. So what's going to happen to the charge on that oxygen? It's going to be very what? Very negative. So these 2 hydrogens, one from glycine and one from serine, are going to stabilize that oxyanion. Okay. All right. So come back up a little bit. So in our next step, histidine 57 is going to donate that proton. So now it has the proton here. It's going to donate the proton to the substrate. So we're going to actually donate this hydrogen to the nitrogen that's in the backbone. Remember NCC, we're going to give it to the N. We're going to give it to that nitrogen, the N terminal. It's not really the N terminal because it's actually a complete polypeptide, but it's on that nitrogen, right? And so we're going to donate that. Well, nitrogen can't maintain 3 bonds. So what's going to happen? You're going to break the peptide bond. That's the peptide bond that you're going to break, right? But in order for that to happen, you have this insanely crazy intermediate. If it's floating all by itself, is this going to require a lot of energy to maintain? Yes. But because we're inside of this enzyme where we have a stabilization of these crazy charges and conformations, we can do this for a whole lot less energy, right? Okay, so we're going to break this peptide bond. This is the peptide bond right here that we're going to break. When we break that peptide bond, now we're going to release this entire side of the polypeptide chain. This is what is going to go out and be released, and that's the carboxy terminus of the protein. But we still have to deal with the N terminus of the protein, and we still have to regenerate our enzyme, right? You can't just break the polypeptide bond and be done with it. So in order to regenerate it, what's going to happen is water is going to come into the active site. When water comes into the active site, our His 57 right here, this is again His 57, is going to interact with one of the hydrogens in the water molecule, and it's going to steal that proton away. When it steals that proton away, now the hydroxyl is going to attack the carbonyl carbon. When that attack occurs, you're going to lose that double bond there, right? We lose the double bond, and we're going to actually cleave our intermediate. So then we can release our amino terminal fragment, and we're going to regenerate our catalytic triad. That's what they call it, our really special setup. Okay, so if we look at our second intermediate, right? So we have, which residue is this? This one is serine 195, right? Our serine 195 needs to be reprotonated. So this oxygen right here needs to take back that hydrogen in order to regenerate back to our initial state, right? What about our glycine already has the hydrogen. Our serine has this hydrogen, so that's good. But this bond will break and we'll be able to release, our carboxy terminus. And so this actually gets kicked out. And when that happens, we now have regenerated our original catalytic triad. It's complicated. It's totally complicated, but it's not a new reaction mechanism, right? You know about protonation and deprotonation, and none of the organic chemistry is new. It's just putting it together. And I'm not going to say in a discussion question, explain chemotrypsin's catalytic mechanism. Go. I'm not going to do that to you. I'm not. I might give you a part of it and say, what's happening? Is this acid-base catalysis? Is this general, or is this specific acid-base catalysis? Is this metal ion catalysis? Is this metal ion catalysis? No, it's not, right? So can you look at it and tell me what's going on? Yeah? I'm not going to make you draw it. I'm not. If that makes you feel any better. I'm not. And I will not use a book that I will not use an image that is not in the book. Okay? All right. Don't get too frightened. Okay. Let's go back. Right. Okay. So we talked about the specificity, right? We already talked about this, that each of the enzymes is not going to be the same. They're going to have a different catalytic triad or whatever mechanism it is that facilitates cleavage of particular substrates. All right. So talking about metal ion catalysis, right? So Nlase is a metalloenzyme. And this is one that you will see in gluconeogenesis. So when we make sugars, not when we break them down, but when we make them. And it actually functions inside the cell as a dimer. And each of the individual monomers, right, contains an active site. And at each active site, the active site has magnesium 2+, which is the purple circle. And then it also has a substrate in it. And so let's look at what that actually is. So if we zoom in on our active site, what we're zooming in on are the residues that are going to interact with the substrate or interact with the the magnesium ions. So here, if we see, do you see this right here? This is the substrate. Our substrate is 2-phosphoglycerate. This is our substrate. Everything else that's a side chain is part of the enzyme. Okay. So tell me about ASP. Tell me about glue. What is GLU? I always just say the 3-letter, but you should be thinking of what they are whenever we say them. So tell me about the charges. They're negative. They're negative. So we have a negative here. We have a negative here. We have a negative here. What about lysine? Lysine's not negative. It's what? It's positive. It's positive. So here you're actually making an interaction, not with the magnesium ion, but with what? What's it pointing to? What is this? It's part of the substrate. So this is ionic with substrate. And I've kind of drawn over, but you can see, do you see the dotted line? The dotted line is showing you what it's interacting with. It's interacting with the substrate. So how about ASP? Is ASP interacting with the substrate? No, these are all interacting with the magnesium. So these act with Mg2+. Okay, how about down, let's see, let's go up. Let's go to the lysine up here. This lysine and this arginine, these are actually going to function as general bases. And we'll see that in the subsequent bases. We'll see that in the subsequent ones. Down at the bottom, the his is actually going to interact with the substrate, which we're going to see. And then this one is actually going to interact with, this is positive, it's going to interact, not positive, negative is going to interact with the Mg2+. So this is just kind of a preliminary look at the enzymatic pocket, just looking at the residues that are going to participate in the interactions. So let's see what happens in the mechanism. Let's see if I can zoom in again. Oh, it is zoomed. Okay. All right. So if we start, right, we have 2 magnesium ions. So what's going to actually happen in this is that we're going to start with one of our lysine residues. Our lysine residue is going to go in and it's going to attack a hydrogen that's inside of our substrate. When we attack the hydrogen, this carbon is still going to want to maintain the same number of bonds. So we're going to convert from a carbon-carbon single bond to a carbon-carbon double bond. When that happens, this carbon, right, that originally is in a carbon-oxygen double bond is going to lose that carbon-oxygen double bond. What happens to this oxygen? It shifts from being uncharged to being negative. So now we have this intermediate that has 2 oxygens and each oxygen has a negative charge on it. That's a lot of negative charges in one place. And so what our magnesium ions are doing are, number one, orienting, because when it comes in, it already has a negative charge. So number one, it's orienting the substrate in the right place. And then number two, once we get to that unstable intermediate, we're going to stabilize the intermediate. Even though it's high energy and it's not very stable, we're going to stabilize it, right? Okay. So now in our next step, so that was lysine 345 that's acting as a what if it pulls off a hydrogen? It's acting as a general base. Okay, so now we have another residue that's going to act as a general acid. So here, down here at the bottom, this, whoa, what did I do? Okay, just hit undo. If you ever get in trouble, just hit undo. So this is glue 211. Okay, so this one's going to act as a general acid. And when it's a general acid, we're going to actually transfer a hydrogen to our substrate. When we transfer a hydrogen to our substrate, what's going to happen to this oxygen? Come down. It's going to become negatively charged, right? So how does that affect the rest of the molecule? If we add a hydrogen here, we're going to do what to the carbon? We're going to lose that bond between the oxygen and the carbon, right? Do you see how we lost that OH? So if we lose the OH, this carbon is not going to have enough bonds. And so this carbon-carbon double bond here is going to transfer between these two carbons. So we just transfer the double bond to the next set of carbons. So if we look here, now we have formed our product. Our product is phosphoenol pyruvate. So we now have a carbon-carbon double bond, and we've lost, right, our hydroxyl there. So it's all about orienting and then stabilizing the intermediate. And that's what we see over and over and over again. All right, I think this is the last one. Feels so bad. Friday is not the best day for this, right? It's really not. All right, so HMG-CoA reductase is really important in maintaining cholesterol, proper cholesterol levels, which is really, really sort of interesting. If you take Biochem 2, it's a big thing because it impacts a lot of people's health and that kind of thing. So understanding this pathway is really, really important. So in this pathway, we take HMG-CoA. So this is the HMG, right? And here's CoA. And so what we want to do is we basically want to cut off the CoA from the HMG, right? And so we do that using a cofactor or coenzyme. A coenzyme of NADPH. The NADPH is going to donate hydrogens, right, to become charged NADP+, and we're going to make melavonate and we're going to make CoA. So basically we just want to cut off the CoA, right? So let's look at this mechanism, last mechanism, I promise. Let me zoom in. Am I still zoomed? Maximum? Yeah, okay. All right. So if we look here, so what's happening? You have a his right here, we have glue right here, and we have asp here, all right? So what's actually going to start our reaction happening? Here's our substrate, HMG, and then here's the CoA, right? So we basically want to get rid of the CoA. Glue is going to act as a general acid. What does that mean? Is it going to give up or take a hydrogen? It's going to give up the hydrogen to the oxygen that's on the HMG-CoA. When that happens, NADPH is going to donate a hydrogen to that carbon. So now we have a tetrahedral carbon, right? And we also have a negative, right? A negative over here. So we have lysine over here. Lysine is going to be positively charged. Lysine is going to help to stabilize not only our substrate intermediate, but also it's going to help keep our catalytic amino acid residues from being destabilized, right? So now we've used up our cofactor. So we need to swap it out. We're going to kick out NADP plus and we're going to pick up NADPH. When we pick up NADPH, right, now this is going to donate a proton to our CoA. When we donate that proton, it's going to break that bond, right? So now what do we have as one of our products? Just CoA with a sulfur and a hydrogen, right? So now you have to look at what happens to the rest of the substrate. So now glue is going to act as a base. It's going to take a hydrogen. We're going to have this oxygen that's going to need to make a double bond with that carbon. So we end up with an oxygen carbon double bond right here, right? And then glue is going to act again as a general acid. And when it acts as a general acid, It's going to donate that hydrogen back, and we're going to get melavonate, and then we'll get CoA. And so we have our two products. Yay, right? All right. So that's perfect. We'll do enzyme kinetics on Monday. That'll be great. Y'all have a very happy Halloween. If you did not get a candy on the way in, please get a candy on the way out. I saw it and I haven't had a chance to respond back to your email, but would you check and see if it's on there now? It should be on there right now. And I feel like, did I miss another one too? Because I feel like I need to really sit down and look at it, because I think there's another one that I'm missing. Anybody need anything on Zoom? Tammy's good. All right. Anybody else need anything? Nope, they're going. Happy Halloween. Okay.